Hello people, welcome back to Molecular Biology and Biotechnology with Lucy. Today we look at the basics of cell and tissue culture. And this is the process by which prokaryotic or eukaryotic cells or organs are grown under controlled conditions. And this was first done in frog neuroblast in 1907 by Ross Harrison. And also Rocks in 1885 had first been able to maintain embryonic chick cells in a cell culture. So the cell cultures, the tissue cultures have been mainly used as model systems for studying basic cell biology, also in studying the interactions between disease-causing agents and cells, the effects of drugs on cells, also understanding the process of triggering of aging, and also nutritional studies, because it's an easier way of manipulating live cells in the laboratory. Also used for toxicity testing when you're introducing new drugs. This is the first level of testing before you go to higher animals. In cancer research, you can manipulate cells, make them cancerous using viruses or radiation, so that you can understand the mechanisms of cancerous cells and also probably the effects of medication on cancer cells, radiation, radiotherapy that is on cancer cells, among others. Virology, we can be able to cultivate viruses for vaccine production like we are doing in the vaccines for COVID-19, and also in studying the virus's infectious cycle to understand how does HIV infect human beings. Genetic engineering, production of commercial proteins like insulin, medicines, and even vaccines. And gene therapy, where we want to replace non-functional genes in cells with cells that have now the functional gene. So we have what we call a primary culture, and now this refers to that process where you get your primary tissue. Here, for example, here you have a lung tissue, and you surgically or enzymatically remove cells from this tissue and isolate them, and then you place them in an appropriate media, and they will grow. Now, once they grow, of course, they'll grow and reach what we call contact inhibition because they have overgrown. And then there's also what we call a process of subculturing, or now getting a few cells from here and putting them in a totally new media, which we call the secondary cell culture. So usually these cells will be heterogeneous in nature, because they are coming from an organ, so they are all different. They have various characteristics. They also have a finite lifespan because they are not able to survive for very long. Then what we usually do is that we do the process of subculturing so that we can be able to continue generating cell lines. So that now you have this one, you can do another subculturing, which you call passaging. Then you have another one, and so on and so forth. But you cannot do this for very long, because at some point the cells become senescent or they cease to multiply. Now, there are cells that you can only have them on primary culture because they do not divide in vitro. That is macrophages and neurons. Now, we have now the continuous cell lines that solve the challenges of primary cell lines. And here we are saying that we will alter our cell lines either spontaneously by having cancerous cells that multiply uncontrollably, or induce them virally, or using chemicals, so that now they can become continuous, such that you can keep passaging and they will continue to grow. Usually these will be small, more rounded, less adherent, and they usually be abnormal. They'll have a higher nucleus to cytoplasm ratio as compared to a lower nucleus cytoplasm ratio in normal cells. Of course, they grow very fast, but have an aneuploidy chromosome number. They have reduced serum anchorage dependence, and therefore most of the time they grow in suspension conditions. We'll be looking at the anchorage and suspension conditions. Of course, they're able to grow to higher cell density, and they have different phenotypes. The expression of proteins in these tissues is totally different from the primary because they keep changing, and the more you passage, the more different your cells become. Now, we have several cell lines that you're likely to come across. For example, human cell lines from breast cancer, from leukemia, human embryonic kidney, HeLa that are used in cervical cancer. We have also those that are from primates, like Vero from the green monkey kidney epithelial cells, and also COS7 from the green monkey kidney cells. We also have others from the hamsters, the CHO, SF9 and SF21 from insect cells, and many others that you're likely to come across. We have different types of cells that is based on the basis of their shape, their appearance, and functional characteristics. We have epithelial-like cells that attach to a substrate and appear flattened and polygonal. For example, those that are from liver, tissue, skin, kidneys. Lymphoblast-like cells usually do not attach, they remain in suspension, and they conform to a spherical shape like blood cells. And we have fibroblasts-like cells that attach to a substrate and usually appear elongated and bipolar. In the culturing of cells, like we have probably mentioned in the previous slide, we have what we call anchorage dependent and anchorage independent. So cell lines derived from normal tissues are considered to be anchorage dependent. Most of the time they'll adhere to the cell media that you have used. Then suspension are usually anchorage independent, and usually we grow them on suspension media. And now those that you have transformed, that is the continuous cell lines, can either grow as monolayers or as suspension. Now cell culture, we have been mentioning about cell culture media. Usually this is a media that tries to mimic the cell's environment in vivo. Then the choice of media, of course, will depend on what type of cell. So we have those that you are likely to come across commonly. We have GMEM, we have EMEM, we have DMEM, and among others. But the most important thing is that the appropriate or general components are that we have a source of energy and other compounds that are able to regulate the cell cycle. For example, a complement of amino acids, vitamins, we have inorganic salts, we have glucose, we have serum as a source of growth factors, hormones, and attachment factors. And we can also supplement our media with antibiotics like penicillin and streptomycin. And once you prepare your media in the laboratory, you filter and incubate at 4 degrees Celsius. We talked about subculturing or passaging. Let us look at it in a little bit more detail. So once the available substrate has covered the cells, we call it a confluent culture. Of course, the growth of the cells will start slowing down, and finally we will have it stopping. So usually what we do is that we need to do what we call subculturing, so that you can keep them healthy and them to keep growing in a steady state. So it is the passage of cells when they reach 80 to 90% confluency, either in a flask, in a dish, or a plate. So for example, we're giving an example here. You've taken a tissue here, and then you have disintegrated it using an enzyme, and then you cultured. This is your culture medium that didn't have, then you cultured, and you can see now this is a confluent culture. It has grown 80 or even more %. So you will do what we call subculturing, so that now you put it in a new media, or you cryopreserve. We look at cryopreservation, which is also a method of storage. So we have 2 methods of subculturing. So we have subculturing adherent cells, and these ones are cells that have attached to the monolayer. So usually we will first wash the cells with PBS, that is free from calcium and magnesium, and then we will add trypsin, dipase, or collagenase in combination with EDTA, and this one is usually to break the cellular glue that has attached the cells to the monolayer. Then we incubate for the plate for around 1 to 2 minutes in a temperature of 37 degrees, and then we shall tap the sides of the vessel, probably it's a petri dish, to dislodge the cells gently, and then we add complete medium to dissociate and dislodge the cells with the help of a pipette, in case there are cells that are not able to dislodge. And then we add complete media depending on the subculture requirement, so that now we can have new growth. If you have a suspension cell, means that it has grown in a liquid broth or media, then this is a bit easier to culture or to passage because you are not detaching the cells per se. So what you do is that once our suspension has reached confluency, then we can aseptically remove a third of the media in the flask, remove, and then you add the same amount, that is the third amount of pre-warmed fresh media, and you can continue growing and you can passage as many times. Now, I talked about cryopreservation or freezing cells. So sometimes when you have cells, you don't want to use them immediately, or even if you want to use them immediately, you can still store them for future references. So we have what we call freezing or cryopreservation, and here we remove the growth media because you can only preserve or freeze the cells as cells. So you remove the growth media, you wash the cells with PBS buffer, and then you remove the PBS buffer by aspiration so that you only remain with the cells. Then again dislodge them with trypsin vaccine, and then dilute the cells with growth media, and then you will transfer the cell suspension in about 15 ml of a conical flask or tube, and then you centrifuge at 200 G for 5 minutes at room temperature. Then you remove the growth media again. Then you resuspend now the cells in what we call a freezing media. Okay, so we have a freezing media. So we are removing the growth media. So the process before the freezing medium is simply to clean them and ensure that we have eliminated all the growth media. And then you transfer them to what we call cryovials. Cryovials, incubate them overnight at negative 80 degrees Celsius. And then the next day, you can now transport the cryovials into liquid nitrogen, like you can see in this diagram. So this is a cryovial. You transfer them inside a liquid nitrogen jar. Now for storage, you can store it for as much time as you need. So this cryopreservation media or the freezing media also depends on cell type, but usually mainly we have DMSO and glycerol. So for animal cells, DMSO 5 to 10%, glycerol 5 to 10%, hybridoma cells similarly plus serum. Plant cells, we don't use the media, we just freeze. We have for animal cells, cell-free and infected, you add fetal bovine serum. Plasmids, you just use glycerol, and phage libraries, you just use glycerol 10%. Now, how do you return cryopreserved cell back to culture? We just do rapid thawing. So the vial that you have used, you remove it from the liquid nitrogen, place it in a 37 degrees Celsius water bath, and keep agitating the vial continuously until it is thawed. Then now in this case, we have to also remove the cryo or the freezing media by centrifuging the vial for 10 minutes at 1000 RPM room temperature. Then you wipe the top of the vial with 70% ethanol. Then resuspend your cell pellet in 1 ml of complete media with about 20% of fetal bovine serum, and transfer properly to a well-labeled culture plate containing now what is the growth media now that you want to have, and check the culture for 24 hours to ensure that they have attached to the plate. And if you find any changes, you can change your media so that the cells are well established. So how do you check for cell viability after you have frozen or cryopreserved your cells? We need to do a process called staining and counting. So we stain using a dye called trypan blue, and this dye usually is permeable to dead or non-viable cells, and it is impermeable to live or viable cells. So you stain and then you load in a hemocytometer here. This is the hemocytometer. You can see it down here. So this is a hemocytometer. You place your sample there and then you place it under a light microscope, and then now you're going to be checking the percentage of viable cells is the number of those that are unstained over the total number of cells that you find in that field times 100%. We also have a problem of cell growth inhibition in cultures, usually caused by cytotoxicity, and it is evidenced by morphological alteration in the cell monolayer or in the cell shape. It can be characterized by abnormal morphology of cells, for example, having giant cells or multinucleated cells, or having a bumpy granular appearance on our cell monolayer, or having the vacuolization in the cytoplasm or the nucleus. The cytotoxicity is determined by substituting materials so that you understand which material used in cell culture is the cause for cytotoxicity. So you can start with the media, substitute serum, supplements, the flasks, etc., each at a time, while you observe what could be the cause of cytotoxicity. We also have contamination of cell cultures that can be due to chemical or biological causes. Now, the chemical ones are caused by endotoxins, plasticizers, metal ions, or traces of disinfectants that are quite invisible, and this makes it very difficult to detect. We also have biological. Usually these will cause visible effects, and they can be caused by biological agents such as mycoplasma, yeast, bacteria, or fungus, or probably there's cross-contamination of cells from other cell lines that you're also dealing with in the laboratory. So what are the effects of biological contamination? We are saying that they are also competing for nutrients with the host cells, and then they secrete acidic or alkaline byproducts that can cause the growth of host cells to reduce or to stop. Then degraded arginine and purine inhibits the synthesis of histone and nucleic acid, and therefore abnormal nucleic acid formation. And then we have them producing hydrogen peroxide, which can directly be toxic to the cells. How do you detect these contaminants? So the first and foremost is appearance. If you notice turbid culture media, there is likely to be a contamination. Changes in growth rates, abnormally high pH, maybe poor attachment. Also, morphology of the cells, are they multinucleated, which could also be probably a cause of cytotoxicity. Granular cellular appearance due to probably fungi, vacuolization, inclusion bodies, and of course death or cell lysis. We also need to do microscopy to observe and confirm if it is yeast, if there is probably chromosomal breakage for mycoplasma, which you can also detect using PCR, ELISA, and fluorescent DNA. We can also do this enzyme amino acids or specific antiserum and monoclonal antibodies or PCR for mycoplasma RNA. We want to amplify and see if it is mycoplasma that has contaminated. Then chromosomal changes, we can detect this by staining the DNA with intercalating fluorescent substances such as Hochst 33 to 58. So what this will do, it will indicate to us if there is any abnormality of our DNA. The best and the oldest way to eliminate contamination, of course, is to discard the infected cell lines directly. Basic equipment used in cell culture, we have the laminar flow cabinets that usually helps us protect our environment as well as protect our culture from contamination. Incubation facilities, we have temperature regulations, we have carbon dioxide requirements, and we have relative humidity requirements. Of course, we need refrigerators because we need to store our media, and we need microscopes to be able to magnify and observe or even check cell viability. And the tissue culture apparatus usually is made from polystyrene. So that is it on basics of cell and tissue culture.